what we have put together is some research project topics, some specialization tracks, and some data types that they could um, go through for these specialization tracks. So it's a matter of fact that when you complete your training program, after that, you review literature and come up with a research question. But if you are a student and not sure what you exactly want to work with, you can explore this page that we have put together for you. I'll just share the link in the chat. All right. So here we are working on 6 different specialization tracks that I wanted to discuss today about. So I showed you some resources that you could learn from, and majorly they revolve around genomic data analysis, transcriptomics data analysis, metagenomics data analysis, or epigenomics data analysis. And then we also have courses on data science, how to use machine learning for biomedical data analysis. And then you also saw the R and Python courseworks that could be used as a tool to analyze these different types of data. So now we know about these different types of data, and we also know about different types of research specialization fields or tracks where these different types of data um, could be analyzed. And we have put together some research questions here under each specific specialization track. The first one is infectious diseases. Simply, it could be you want to work with COVID data. You have some questions put together here under different types of data analysis that you can pick. You can take your time to read through this page after this session is over and let us know what specific question or field interests you the most and what you want to work on. All right, so back to infectious diseases. Infectious diseases are simply communicable diseases like Ebola or COVID, or also we have some case studies put together on the portal in the example projects that you can go through to get an idea. And we have the example projects linked up here as well. If you want to go through them and see how you can work on these topics, you can go through. The next specialization track that we have is oncology. So here we have various types of research questions that you can answer under different categories of data analysis that is possible. And the list is very huge in number. You can go through each one of these different topics and pick the one that interests you the most. And here we also have the multi-omics integration possible. And also researchers and research fellows have been working with us on structural biology and cheminformatics questions too under the field of oncology. Again, we have the example projects listed out here for you. And the third specialization track that I would like to mention about is precision medicine or personalized medicine. We are currently working in this domain with transcriptomic data, but yes, also there is a possibility that you can work with clinical data, epigenomic data, or genomic data under this category. And then you have agriculture as another specialization track, and then different types of data analysis possible under this specific track. Neurosciences is another interesting specialization track that you can pick. And we have been working extensively on Alzheimer's disease, Parkinson's disease, and also other neurodegenerative disorders, as neurodegenerative disorders are among the most researched topics. So this could be one of your research topic that you would like to work on as well. And the fifth specialization track is metabolic disorders that could be answering some questions related to diabetes. Diabetes type 3 is a topic under research now, and we have few researchers and research fellows working on diabetes extensively. So if you want to join them and work with them in a group, it is possible. And yes, definitely working on the individual research project is a possibility. And the last specialization track that I want to mention is space omics. Now, this is one of the most interesting fields that is evolving. And we have some amazing space omics research projects that I would like to definitely take you through. I'll just show you these projects that you can go through and see what is the possibility for you to work on such a project. Again, I'll repeat, the link is with you in the chat box. We'll also share it with you over an email. You can take time to go through each one of the research questions or topics that we have mentioned under each type of analysis, under each specialization track, and think of what you would like to work on. And that was it. I did not take up each one of the questions because it would have taken more time. All right. Now the question arises that we have talked about different types of data analysis and specialization tracks. But where do you run these pipelines that I was just mentioning about? I discussed about algorithms, and where do you have all of this data analysis possible? So the answer to that is the T-Bioinfo server. Now, this is the T-Bioinfo server, the hands-on practical server that you will be using to run all the different types of pipelines for different types of analysis. Here you have all these analyses put together in different sections. You have the transcriptomic data analysis section, then the genomic data analysis, metagenomics is here, data mining, and then you have some phylogenetic analysis possible here on the server as well. I can see a question in the chat. I'll just take up in a minute. Or I can answer it now because it's related to the specialization track that we just discussed. So Gibson is asking, every research specialization is interesting. What are the key criteria to consider when deciding which research specialization to go for? Now, the criteria is definitely, the first one is your interest. What is your interest? What you want to work on? The second one could be in which field you want to do your majors. If you're a graduate student and you want to pursue your master's, it would be in which field, or a PhD degree, then in which field? What are you interested to learn about? Because it is more important to streamline your interest with the learning. And if you don't have an interest, let's say, if you don't have an interest, though that's a very big thing, but still, if you don't have an interest, you can maybe pick up one of the research questions that is more under, I'll say, curiosity of the researchers and they're working on, and you think that you would like to add on to the research society by working on that topic, you can pick that. But again, it's the interest that you want to work on and where you see your career going, or where you see your majors, where you see you pursuing your majors into. I hope that answers your question. All right, so now getting back to the server, where I was just showing you the different areas of analysis and different sections on the server. So here you have these drop downs, which will take you to some demo pipelines. Now, before you join a program, if you want to see how the server works and how these different pipelines are arranged, you can create an account on the OmicsLogic Learn portal, and that will help you get an account on the server as well after 24 hours. And then with that account, you can access some of the demo pipelines and see how these pipelines basically work. I will just show you one simple pipeline today, and it would be a very brief pipeline, just to showcase how interesting it is, and to how interesting and easy it is to run a pipeline on the T-Bioinfo server, and how interactive the results look like. So here we have a demo pipeline on breast cancer. And this is a demo pipeline where the data is already uploaded at the backend, and we'll just run the pipeline and see what the flow of the pipeline looks like and what the results look like. So I'll just give you a background of what this pipeline is. This is a breast cancer pipeline where you have 10 patients of different types of breast cancers, I'll say. The first 5 patients are ER positive breast cancer type patients, and the other 5 are triple negative breast cancer patients. Now, I am interested to look what are the different differentially expressed genes among both of these groups, and also then know about the pathways in which these genes are involved. So let's begin with this pipeline. Yes, so there are no prerequisites actually to perform the data analysis, Kishore. And it is not important that you have a software background to run a specific pipeline. We'll be teaching you everything you need to know about a pipeline, what type of data you upload, how you run the pipeline, and how do you interpret the results in the training process when you join the research fellowship program. So you need not have a software background to do so. All right, now getting back to the pipeline, I start the pipeline, and then to look for the differentially expressed genes, I click on the DSEC2 option, and then to look at the pathways involved, I will select enrichment analysis and GSCA and simply end my pipeline. Now, when I end my pipeline, as I told you, it's a demo pipeline. We already have our results put together. And now I'll quickly open these results to show you how easy it is to understand what the results look like, interpret biological insights. Now, we have a list of results that are put together here. We can download these zip folders and extract the files and look at the differentially expressed genes. We have some very interesting plots, and we also have some very interesting TXT files put together where we can see the value of each differentially expressed gene for each and every gene with regards to the samples. So this is a volcano plot that you see. Now these plots are interactive. You can see which is the gene that is differentially expressed, what is the log fold change, and these terms that I'm using. It's not important that you understand it now. We will definitely take you through the training process where you learn about it all. And this is a heat map which clear cut shows you what is the expression level of different types of genes in different samples. And more the expression level, more different is the expression level, highly is the differentially expressed genes that are mentioned here in the heat map. And then you have some interactive dashboards where you can interact with these results and also interpret what the results look like. All right, so you can play around, you can increase the fold change values, and then look at the differentially expressed genes. And also, if you want to look at the pathways, then you can click on the enrichment button right here, and you'll get these pathways along with the gene ratio, and you'll get to know about what all are the pathways involved here. So that was a very brief overview of how you can run a pipeline, and just to showcase you how easy it is to run this pipeline and then interact with these results. All right, now my last question is, what after research fellowship program? So one possibility is definitely, as Ruthvi has shown a publication as well and told you, it is the publication that you can get if you dedicate good number of hours to the research fellowship program and work on this program. So here are some of the publications that we have got by research fellows, and you can go through these publications. Here is the link in the chat box. And another outcome could be a publication on the OmicsLogic Learn portal as well. Now, there are many students and researchers who work on a short-term project maybe. And so what they do is they submit their project once it's completed on the portal. And then when they extend their fellowship, they validate their project, and then take it towards the publication. So we have all our projects here uploaded under the projects tab on the OmicsLogic Learn portal, and you can go to the portal and look at some of these project publications to see in which all different specialization tracks and with which all different data types our research fellows have completed their research projects.